Hi guys. Let's see.
So you put how that
I did. It just say like there's a few matches. Thank you. Appreciate it. Yeah.
Yeah.
Okay.
So
Thank you.
unless you want to walk that.
No, I didn't want to pack all my stuff.
Welcome guys. It's just gonna be a minute until we start. We still have a couple minutes. And yeah, everything's backwards. So appreciate this welcome. I had to write it backwards. So but we'll get started soon, okay? For those of you guys who are here, thank you guys. Could you guys go ahead and put in the chat your first and last name, and then I'll take your ID number, your @00, and then your number. And then this is everybody's first course, or there's everybody's first session, so I'll just write yes for that. But thank you guys in advance. Right, I'm gonna finish up writing this down just to let anybody else trickle in. But yeah, looking good so far. Can you guys hear me okay? I'm not too quiet. You're not having to put your volume all the way up. Everything's fine. Alrighty. Perfection. Alrighty, let's go ahead and get started. So you guys know me, I'm Lauren. I wanted to just do kind of a brief introduction because I mean, you guys are just like, okay, cool, she's here, kind of thing. So I took micro last semester, and as Dr. B already said, I passed with an A. I can give you guys a lot of tips and tricks per my contract. I cannot give you any answers. Can it puts me liable, and I just can't. I'm sorry. I know that you guys are going to be, practically begging me at some point that, it'd be so much easier if you just tell me and I can get on with my life. But yeah, I'm sorry. Um, so there's me, and then there's also Caesar Delgado. He's another pal for microbiology. He's more towards Melina, but if you guys can't make my sessions for whatever reason, we can see if his availability is more open. Um, I think I mentioned in class and online that my sessions are going to be Mondays at 5. They will be at on Wednesdays at 7. I'm gonna double check that because I feel like I'm wrong. Wednesdays at 7, and then I have one short one at 11:30 in the morning on Thursday. So those are my 3 sessions. Of course, you can also, in the Remind app, you can do a private discussion and ask me questions. And I can try to give you the best response, or I can say, hey, if you can make it to the next Zoom meeting, then I can review it there. I'm in a special area right now where I have a whiteboard, and I'm grateful for this whiteboard because I can actually draw things out and show you guys diagrams and things like that. So if you guys have anything you want to pick my brain about, either about me, about micro, by all means, you can either unmute yourselves, type it in the chat, whatever you'd like to do.
So for tomorrow's quiz, is that just going to be on the syllabus content, or will it be like around chapter 1? Or
so it'll be like a syllabus quiz basically. It'll have a lot of policies. It'll be like like certain things like, oh, when should I work on my micro project? As soon as possible will probably be the answer. There will also be things like the biosecurity levels that you guys probably talked about on day 1. So those review that, anything that's in your syllabus, basically. Anything that you've learned like this week about the plates and things like that, not going to be on it.
okay, thank you so much.
Are there any other questions, or like I can go over the streaking technique if you guys are confused about that, or if you guys want to kind of know why we do it. Honestly, pick my brain. I'm here for you guys. Yeah, I could totally go over that. So let me get out here. Let me erase this. Okay. So I'm so glad I have this whiteboard. I cannot tell you, I am so much of a drawer when it comes to like learning. So I always like to have something like that. So we have our plate, right? That's a really oblong plate. Can you guys see that okay? Okay, perfect. So we have our plate, right? And for reference, this is going to be our auger side up, right? This is our auger side, not our lid. So when we look at our auger side up, we're gonna want to first make our true false, right? We're gonna make our true and then our false, right? Of course, if this is our TSA plate, then we're gonna write probably TSA, right? I'm gonna put my I like to do my first initial last name. So I'll put L. And then I'm also gonna put the date. Today's date is the 24th. And then it depends on what type of what's going to be on our plate. So today in lab, if you were Monday, Wednesday, or on Thursday, if you're Tuesday, Thursday, you guys or I'm sorry, no, Monday, Wednesday, you guys did this, but Tuesday, Thursday, you're going to be doing this tomorrow. So this, I guess this will just be a little bit of a precursor for you guys. But we're going to take our environmental swabs, right? So I swabbed the sink. So I'm going to write not only what type of auger it is, it's going to be our TSA plate. It's also going to be my name, date, and the samples. So my sample was the sink, and I'll write that on here. Alrighty. So this is how we're gonna set up our plate, right? Auger side up. We're gonna put all of this information on it. This is what it is. Cool beans, right? Nothing too. We're just getting organized, right? Now comes to the application part, right? So we're going to start with our quadrant 1, right? If you ever get mixed up on these quadrants, what I used to do and what Dr. B does is I would put 1 somewhere in the quadrant, I would put 2 somewhere in the quadrant, 3 right here, and 4 right here. So it's going to go 1, 2, 3, 4. Okay? You can write it really small in the corner. You don't want to try to do anything like smack dab in the middle, because this is where we're going to try to see our bacteria, right? So our bacteria is going to, when we're in the first quadrant, it's going to be really thick, right? Because we want to get a good population going. And then through the plate technique, we're going to kind of sparse it out to see what it looks like. Kind of, like if you look at a big field of corn, you're gonna be like, well, that's a big field of something. But if you go up close and you take a corn off its, stalk, you're gonna be like, oh yes, this is, whatever, white corn. I know what this is. I can see it, and I can say, how many kernels it has if I really wanted to count it out. I could say, oh, it's this long. I could say, oh, it looks shiny, right? I can say it's, firm, whatever, right? So those characteristics are what we're looking for. So we'll learn about it again in another selection, but I guess I could preface it for you guys. It's going to be called CBEST. It's going to be your best friend. You're going to use it now, and you're going to use it on your unknown report. That's the 26-page one. So this CBEST, right? Let's see. Oh, I can write it. The CBEST, we'll just write it like this. Now, what is the CBEST, right? So C stands for color. We're gonna find out that, we're gonna find out that bacteria can look kind of like a yellow, or it can look a little more white. Sometimes it can look almost translucent, right? We're gonna try to look at the color and describe it, right? And so that'll be our color description, right? Then we're gonna look at borders. So borders are gonna be, is it circular? Is it, a little more irregular? Like, what does it look like? You're also gonna learn about a special thing called swarming cells. Now, those are gonna be fun. And I promise you, it's nothing too complicated, but it is really interesting, okay? But those are just some of the examples that you could put for the border, right? Now let's go on to elevation. So this is why pictures don't really do plates justice. For elevation, you're actually going to turn your plate kind of sideways. You can still have it closed, but you want to look at how it looks over the auger, right? We're going to have our auger And if we have something very flat, a bacteria that grows flat, it's going to just lie on top of the agar. You can hardly see any elevation, right? And then we're going to look at things that maybe are just rounded. They're nice little domes on, right? And there are special words for these. You'll review them in the next class, but I'm just trying to give you guys examples. And then there's going to be, you'll laugh about it in class, but there's going to be a nipple one. It's going to look kind of round and then perturb a little more and then back down, right? So those are going to be our elevation. That's our elevation, right? So moving on to the size. So do you guys remember those little rulers in your lab drawers? So those little rulers are going to be used against a guide to help you figure out what the size is. So obviously small, medium, large. But then we also have our special one. It's called punctile. If you guys have taken like medical terminology or you know your way around words or like prefixes and suffixes, punctile means just barely anything. It's a very small, it's smaller than small, right? So that's going to be what we would put for size. And, if you guys want me to, I can, I can say, okay, well, this is what this bacteria would look like, right? And I can see best it for you. But we'll get to that later, okay? I'm just trying to write this out so that you guys can maybe go into lab and feel a little bit more prepared. Alrighty. And then finally, we have texture. Alrighty, for texture, it's just what texture is. Is it going to be dull? Is it going to be shiny? Is it going to be matte? Think about spray paints. Like, what is it going to look like? So, like, obviously, this whiteboard, you can see the reflection on top. That's shiny. This is a shiny surface. So this texture is shiny. When you look at your plates, you can use the lights up in the lab and you can see it. You'll be able to see the texture. But that will be our C best for what comes out on this plate. Now for the actual streaking technique. So for this, I might, scribble over my writing already, but for the first quadrant, like I said, we want to get a nice thick lawn. So we're gonna just get all up in there. Man, that is not the best expo marker. We're gonna wanna, get up all in there. Go back and forth. Just cake it on, really. We want to get the highest bacterial load that we can, right? So that we did that with whatever sample we had, right? This was my environmental sample. So then we're gonna move on from our environmental sample. That's out of the way. I'm no longer gonna touch that. I have my inoculating loop. I'm gonna have, this is what's gonna go into the fire. So my inoculating loop is ready to go, but it's dirty. It's been in my drawer. So I'm gonna have to stick it in my incinerator, right? And if I'm going too fast, by the way, let me know if I need to stop. I know some of this stuff is, we just went over it, and some of the stuff you guys have yet to go over it. So feel free to stop me. But I'm gonna stick this in my incinerator, right? Once I stick this in my incinerator and let it cool down in the air a little bit, I'm not gonna go touch people. I'm not gonna put it back down on the surface and get it dirty again. And I'm definitely not gonna go to my auger, because what'll happen is if your loop's too hot and you touch the auger, it's gonna sizzle, right? And we don't want that, because think about how you kill bacteria. One way to kill bacteria is to make it really, really hot, right? So that's what our loop is if we just touch the plate after putting it in the incinerator. So I have let's say my loop is, ready to go. So my loop is ready to go. We're gonna go, 1, 2, 3, 4, 5 in there into that first quadrant, 5, right? And I'm still keeping my loop on here. And then we're just gonna go back, and just coat it up, right? So 5 times in here. So this is gonna be, this is gonna be 5 times. We're gonna enter this one 5 times, right? And this was our original lawn. Alrighty, now my extra marker is dirty again. My inoculating loop is dirty again. So because I just touched a bunch of bacteria, I'm gonna have to put it back into my incinerator. Waiting. It's gonna turn orange. It's gonna be ready to go. And let it wait. Gotta make sure it's not gonna burn anything. It's not gonna kill our bacteria before we can grow it. Alrighty. So now she's ready to go, nice and cool. Now, you know what? Brief intermission. This is my clean. This is my clean inoculating loop. I just want to do different colors. So now we're gonna do about 2 or 3 times into this one. This one is, this is gonna be spacing it out a little more. So we're gonna go 1, 2, 3, and then back and forth. Alrighty. So this one is gonna be 3 times. All good? Alrighty, so 3 times there. My inoculating loop is dirty again. I just touched a bunch of bacteria. Stick it in the incinerator, right? Gotta get hot. Let it wait. Waiting. Now this one is gonna be what we're gonna separate out the bacteria. This is how we're gonna get our sea best, right? So how we get our sea best is by looking at the colonies in this quadrant. So this quadrant, we're only going to go 1 or 2 times back into 3, right? So we're going to go 1, 2, that's it. And then we can do a little squiggle, and then we do our Harry Potter lightning bolt, right? And then we can put our inoculating loop back into the incinerator. So that's clean. Remember, this is bacteria. You want to make sure that this is as clean as possible, but also everything around you. You definitely don't want to take any of these things home. So stick this back in the incinerator. It's clean. We're gonna set it aside, right? So then we're gonna put this into the incubators, and Monday, Wednesday, you guys did that today. Tuesday, Thursday, you'll do it tomorrow. But you guys will put it in the incubators, and next lab period, you'll be able to see what grows. So this is very important for your environmental swab. You're seeing how nasty it is. And I'm gonna be honest, there is unimaginable things everywhere. So this is why I wash your hands all the time. But this is definitely it's going to be an exciting one. And then also I wanted to clarify something right here. So this sea best, it's going to go by another word other than just sea best. It's also going to be called colony morphology. Alrighty. So colony morphology. That's going to be the sea best. See, that's good. But yeah. So any questions on any of that? Did I go too fast? Remember, this is my first time doing this, so if I'm going too fast, let me know. I will take constructive criticism. Don't worry. Okay. Perfect. Okay. So is there anything else maybe about syllabus that you guys want to know? I don't have it in front of me, but I could definitely pull it up. And then also, is there anything like are the biosafety levels, are they tripping you up? Or is there anything else you want to know about studying tips? Like, I'm here for you guys. You guys can take pictures of this. I can get out of the frame. But yeah, can you guys see the bottom too? Can you guys, let me see. You guys can see it. Okay. Alrighty. So that's gonna be your colony morphology and CBEST. I know I've been talking for a while. Let me also put on here real quick. What study techniques? Okay. Oh yes, I will also post the recording. And yes, I'll always post the recordings. If you have the Remind app, I'm gonna just see what works the best. But I'm probably going to send it out as a reminder and say, hey, under the files, you'll be able to see the review sessions that I recorded. And I'll probably do that. I'm just gonna have to see what works the best. But yeah, I will get that available to you guys whenever you guys, if you guys do miss a session or anything like that. But yes, and then I also got a question: what study techniques helped me? My thing was that I had to find what worked best for me. So one thing that worked for me was that I did have to go back and rewatch those hour-and-a-half lectures. I know it's really daunting and it's gonna suck, but it was the fact that I couldn't keep up in a lecture, that I fell behind and I felt like I wasn't getting it all. So you can be there to ask questions and get on the spot responses and explanations, and then go back and be able to go over the information that you might have missed. So that was one of the things I had to do. Another thing that helped me with at least studying, I made a lot of Quizlets. There's a few, there's like a few units where you're going to see a lot of memorization. And when you have to memorize a lot, the Quizlets help. There's a test mode on Quizlet that I absolutely love. But in all, like, it's just finding what works for you. If you're more of a visual person, do what I do and draw it out. You know, if she draws a diagram in lecture or in class, you draw it out. You know, it's if you're more tactile, you guys need kinesthetic. That's the word. If you guys need something like that, try to make a model. Try to look at models. Try to see if you can go to Dr. B and talk about it. I talk with my hands sometimes, and so what I like to do is like like I'm streaking for isolation, right? And then I play it out kind of. If you're a more verbal person, sit down and get kidnap your friends and sit down and say, I'm gonna lecture to you guys about different, like you're gonna do antimicrobials later. You'll do a bunch of stuff later, but yeah. Another question for the quiz, did she say we needed to memorize the different examples of organisms from the biosafety levels? So you do not need to organize what the specific organisms are. Like, you don't need to know that E. coli K12 is BSL 1, but E. coli OH 157 is BSL 2. You don't need to know those. All you need to know is the exact wordage, right? Or the words she uses, right? So I know that between two different levels, it says something like potential to be hazardous or aerosolized. Memorize what the descriptions are for those, because the wording can get tricky on those tests for the quizzes. More likely than not, it won't be on the exam. So maybe the only question on the actual exam itself will be like, what level is this class? And you'll say, what level is this class? Oh no, my battery. Yeah, good job, good job. You guys are good. honestly, you guys showing up and wanting to ask questions is really good. Um, like this, being very prepared for, especially the first exam. That was the one that I scored the lowest on because I just had to figure out what worked best for me, and I just didn't know how to study. So, it's going to be a little bit of trial and error. If you see that if you're taking a quiz and you don't quite understand maybe what's going on, or if you're having a lot of time struggling, or if you get your quiz back and you say, "Oh, I didn't do so hot," then you'll probably want to go back to see what you did to study. You'll probably want to try to change that, and also, you know, did I go to all the lectures? Was I paying attention in class? I think one of the main reasons why I probably didn't do so hot on the first exam was because I actually fell asleep during a class. Yep, I was conked. I don't know what I was doing. I was probably staying up too late, but um yeah, it's just finding the balance and doing what works for you, trying different things and figuring it out. Are there oh, oh yeah, no problem. I'll see you in the next session. Alrighty, it's about 5:30. I will be here for an hour if you want to pick my brain about anything. I know the first section is probably going to be a little dry. But let me know. I'm gonna go ahead and let you guys take a 5-minute break. So we'll be back 5:35. You guys can take a quick 5-minute break, get a snack, or look through your notes to see if you guys want to ask any different questions. So I'll go ahead and stop my video and mute myself, and you guys can go ahead and do whatever. I'll be back in 5 minutes. All right, guys, we'll be back. All right. Let me see. From the first lecture, do we need to specifically remember each scientist and what they discovered or the experiment they performed? The big guys are going to be Louis Pasteur, right? And what he was known for. You're probably going to want to know the father of microbiology, the inventor of the microscope, and kind of his story. But I would just say big ideas. I can hardly remember a lot of those guys. I mean, I know Leeuwenhoek, I think is his name. That's somebody you're gonna want to remember. But Pasteur definitely, Dr. B has a little bit of a crush on Pasteur. So you're probably gonna just want to know what was reviewed in lecture to a certain extent. You don't need to know that. I think it was Leeuwenhoek, who was the guy who made the microscope. Correct me if I'm wrong, but I'm pretty sure the story is something like he's somebody who made eyeglasses, and he wanted to create something so that he could, see better or something, see something better. I forget what exactly, but maybe just like the story, but nothing too deep like dates. You will never need to know any dates of history figures. But yeah, I mean, if she reviewed it in lecture, I would definitely make sure that you at least know about the person and you can make inference clues about the person. But yeah. Were there any other questions about, I know it's like not a whole lot of information, but as we get going, there will be plenty of opportunities to see what we have in store and what questions can arise. But you guys don't need to stay here. If you guys are good to call it quits, then so am I. If you guys don't have any more questions, but I will stay available until 6 o'clock. Okay, so if you guys want to peace out, you can. Do I recommend having the lab note manual for the class? I'll start with that one. So there's a lab manual and then the lecture manual, right? The lecture manual is nice. It's not the most helpful, in my opinion. A lot of the time, if you cannot find it in lecture, it's not going to be on exam. So when you go through the lecture packet, it's going to be a lot of fluff. It's going to be what dates were these. It's not going to be a whole lot of like information that you need to know. So what I would do is just focus heavily on what she says. If she doesn't mention it, it's not going to be on the exam. There's no material that you don't know about, right? So that would be the lecture manual. The lab manual, you'll 100% need. Every single day you come into class, you'll need that. It's going to have your instructions. It's going to have places where you're going to fill in the blanks, and then you're also going to have to actually upload those and submit those, right, for the lab assignments. So there's that. And then I have another question. Are the quizzes based on the lecture note or the lab manual? So neither. Well, I mean, the lab manual is going to be the lab portion of the exam, 100%. Anything covered in lab is fair game on that lab portion. But on the lecture portion or the written portion of the exam is going to be solely what she records in the morning. So I would definitely recommend going to those lectures in the morning. I know it's hard to get up, but like I said, if you go there, you hear something, you ask questions, it's going to be able to engage you a little bit and you'll be able to retain it a lot better because it'll feel like a conversation, right? And then if you were to watch a recording, it's almost like trying to watch a YouTube video. Sometimes it's helpful, sometimes it's not. So if you're one of those people who cannot take fast notes for the life of you, I would recommend also copying down the slides before you come into class, listening to the lecture, engaging in the lecture, and writing notes on your notes, right? So if writing fast something you struggle with, then I mean, my hand likes to cramp a lot. So I would try to write it before I actually go into lecture. Let's see. Since you split up the lab assignments, do you need to study the sections that you are not responsible for as a way to prepare for the quiz? Let's see, or the test. Okay. So yes, you'll need to at least review those. So there's going to be things like labeling a graph. You know, you need to know if you weren't necessarily responsible for that on the lab, you'll need to know how to make a graph. It's good to at least engage in a conversation with the people who did those sections and for them to let you know kind of, you know, what they did, how they came about that, and you know, at least understand those types of concepts. There's one, I will preface this now. Don't be like me. I if you go to lab 15, you don't need to right now, but if you go to lab 15, it's all of the parasites that you'll be learning about, right? So lab 15, I did my parasites, right? I was the helminths, the worms. So I did my section, but the test was going to cover any of those. I didn't decide to take a deeper look at those other ones. So at the last minute before the exam, I was struggling. I was like, what are these parasites? I had to retake the parasite notes like 5 or 6 times because I was trying to drill it. I didn't do my own research or just like look at it because I'm pretty sure on that one you're gonna be like given something like, you know, let's say it's tapeworm. That was what my micro project was about. So tapeworm, right? What kind of issues does it cause? What's the duration? Where is it mostly found in? You know, things like that. I knew everything about worms, but when it came to anything about flukes or something like that, you know, anything else about other parasites, sorry, anything else about different parasites, it was kind of up in the wind for a little bit. So it definitely at least take a look at other people's stuff. You know, if they're being uncooperative, that's one thing. But if they're going to be able to engage with you, then I would ask them all the questions that you can because it's not only going to help you learn the information, it's also going to help them retain it because the best way to learn something is to be able to teach it, right? The best way to be able to, you know, store this information forever is to be able to teach it. So good question. Definitely because you're just like, less work on me. This class is a load full, right? I don't need to worry about it. But there's a little bit of worrying that you need to do, unfortunately. It's a 900 point class. A lot of other classes like Bio 32, Bio 33, 600 points, right? Max. But this is a 900 point one, but you'll find that it comes pretty easy, you know. But if you're a bad test taker, I'm a bad test taker. It helped me because I was able to do the labs, understand the content, and do well on that portion of the exam. But when it came to the multiple choice, I don't know what's wrong with me, but I bet it every time. So whoa, train of thought gone. Sorry. Um, Back on the rails. It's getting there. Can you tell that I'm spacey? But anyways, so when you turn in those assignments, they add up to a lot of points quickly. When you turn in all 21 labs, it's a lot. When you turn in the case studies, it's gonna do great things for your grade if you're a bad test taker. If you strive to do the best on those, you know, one lab is worth like 10 points, right? But if you do, let's say you didn't do like 5 labs, but you got 100% on one case study, you know, that's the equivalent is 5 labs to one case study. That's the same amount of points, right? So do well on assignments, and you'll in the process also do pretty well on the exams. If you're a bad exam taker, then it'll help cushion you a little bit. But yeah, one thing I did want to mention was that Dr. B was talking to me about how I take exams, and she said it's a really interesting and thorough way to try to get to the answer. So what I do is I take a question, right? So if it's something like, let me try to find an example. If it's like, what does the B stand for in CBEST, right? If I get asked that question, then I will write out CBEST, color, border, elevation, size, texture. And I'll be like, okay, B is what I need, border is what it is, right? So that's kind of what I do, is that if there was more than one step, I'd be able to write it out. And also sometimes you just space out during an exam. You freak out and you're like, I forget what CBEST is. But you wrote it on the lecture portion when you go to take your lab portion. So you're just like, oh, okay, I'm good, right? You're like, oh, I remember writing that, you know? You can get frazzled very easily. One of the things that would happen to me is that my heart would start pounding and I couldn't get it to stop. So controlling those breathings, get into the right mind set, practice mindfulness. You know, if it gets too much, one of the things I always say is that you can't go chopping down a tree with a dull blade. Also, yes, you'll be able to write things out for exams. You can write it in the margins, and you can also ask Dr. B for an extra piece of paper. She just has to make sure it's blank, okay? But chopping down a tree with a dull blade, right? So when we're in our Zen moments, we're sharpening that blade. So we can get back to knocking out all of those trees, all of those assignments. So that is my probably best advice, is that if you are just trying to put your head down and do it, that's great if you have determination. But if you're just like, I need to get this done, there's a lot writing on this, I'm so stressed out, it might take you 30 minutes to take the exam that way, or 30 minutes to do an assignment that way. But if you take 5 minutes to breathe, it may only take you 10 minutes to do the assignment. So it's going to take you half the time if you take a break, Like, I'm sure you guys this isn't like your first semester ever at BC. You know that there's plenty of resources here to help you. There's not only me, but if you feel it necessary, you could get your own personal tutor. But you also have Dr. B. You can reach out to her. She has her office hours. Utilize them. It's a great resource. But in all, yeah, it's just kind of doing the same thing you guys have been doing, just working it out, figuring it out. Just a quick question. I know I didn't get to ask you guys this, and I don't have your guys' responses right in front of me. But what are some career goals for you guys? You guys can unmute yourselves, type it in the chat if you feel more comfortable, whatever you'd like to do. RN, yep. RN, got a good one? Are we gonna go for 3? All right, well, nursing is a good, yep, RN. I'm definitely trying to go down the nursing pathway. I'm currently in the CNA program. So be patient with me. If I'm really tired on Wednesdays, those are my clinical days. But I will be as preppy as possible. I will chug a bang before I do my 7 p.m. reviews, okay? I'm here for you guys. You guys ask me questions and I will answer them. And if I can't answer them, I'll get Dr. B to answer them. You know, if you feel intimidated by the whole Dr. B, or if you feel intimidated because she's your professor, don't be. She doesn't bite. I don't bite. He bites. I mean, we'd probably be in an institution if we did, but um, we're fine. We're humans and we understand. If you're stressed out and you're like, I don't understand this, and we see it as a very obvious answer, we're gonna be like, take a deep breath. What about this? We'll ask you questions so that you can, it leads you to understand. And you're like, oh, okay, yeah, that totally makes sense. Why didn't I see it that way? Um, we're gonna try to help you guys learn, and we're not just gonna tell you the answer because it takes away from, like, you work so hard to figure it out. And if we just tell you, it's like, oh, I should have known that. But if we're like, well, what about this? And then you figure it out, then that's your aha moment. I want you guys to experience that. I want you guys to be happy that you figured it out. It takes a lot of brain power. But yeah, if you have any questions about me, I have dogs. I mean, if you're a dog person, I love dogs. I miss my dogs right now. But if you have any other questions, you can type them out. I'll be, again, we're almost going to the 6 o'clock mark. I'm happy. I'm good. I just hope my computer will last that long. She should. It should die right at 6 o'clock. But yeah. Also, depending on how the semester goes, I might actually be able to do something in person. So if you guys are interested in that, you guys can shoot me a message either now, whenever, on Remind. I'm just gonna try to see and get a feel for if people would like to have that happen. Yeah, I know I learned better in person. I was so glad it was in person last semester. And you guys are very lucky this semester. I mean, I hope it continues to be more in person, but yeah. I found a nice little nook with whiteboards and seats. Like, I can have a couple other people in this room. It would be great. But yeah, we can do something in person. I'm a stickler for masks, unfortunately. One other tidbit about me is that my dad is actually immunocompromised, so I don't want to take anything home to him. I am very anal about making sure everybody washes their hands, everything's wiped down, everything's sanitary. I have Lysol wipes always on me. But yeah. So if you guys, oh,